So let me tell you a few words about how all this started, because some of you are with us from the beginning, but some are more novel. And actually, the idea was born in 2016. We had something we called the Glyco-IgG brainstorming meeting about the glycosylation of IgG. And I believe it was Claudio Franceschi, I'm not sure, maybe somebody remembers, who said, maybe we should start the human Glycome project. And then I liked the idea, and we said, okay, let's start the human Glycome project. But then we realized that the same idea was born in the US, and the Recoming started the human Glycome project in the US, and then we said, okay, we are not going to have two human Glycome projects. So instead of having the human Glycome project in 2017, we had a meeting which we called the Human Glycome Project Initiative, because we were negotiating with Americans to merge the two events. So although we did talk about the human Glycome and brainstorming what to do, we didn't call it the Human Glycome Project meeting. And this took place again in Dubrovnik in 2017. And then until 2018, we did negotiate with American colleagues. We merged the two initiatives and had the first official Human Glycome Project meeting on the October 6th, 2018 in Dubrovnik. And at that time, we already had 33 individual projects included, over 200 participating researchers who kind of tried to align their research ideas and plans into this Human Glycome Project meeting. And we're really making good progress. We met again in 2019. We had this organized together with the 11th ISAPS conference in Split. And then came the pandemics. And the pandemic stopped so many things. We haven't met in 2020, we haven't met in 2021. And finally, we managed to meet again in November 2022, actually a little bit more than 6 months ago. And this was in this very same hotel, and I believe most of you were here. And now we are meeting on the 4th meeting of the Human Glycome Project. Its project is formally 5 years old now. We do have some fantastic results. We have more plans, and this is what we are going to discuss in the last few days. But before I move to science, I would just like to tell you a little bit of history, which is actually quite amazing in this part of the world. So in a little bit less than 1800 years ago, there was a very powerful Roman emperor called Gaius Aurelius Valerius Diocletianus, who was actually the only Roman emperor who retired. Everybody else was killed on duty or died. He decided to enjoy his final years of life and retired in 305, and looked through the vast Roman Empire, which was nearly at its peak at that time, and decided to build his palace here. So this is the Diocletian's palace, the way it was drawn a long time ago. And this is when you look at the aerial photo of Split today, you can still see parts of the original palace walls and some of the towers, which were part of this palace. So if the Roman emperor decided this is the best part of the Roman Empire to retire, it's also a nice place for us to come, and I hope you enjoyed both the region and this hotel, which is one of the best business hotels globally. So most of us are from the glyco field. We understand glycans a lot, but unfortunately, we are the vast minority globally. Most scientists still ignore glycans. So the protein you see in the middle that says glycoprotein, we see how much glycans are in the surface. We cannot understand the function of S-glycoprotein without its glycans. But many people try to do it. And the best analogy I make, and I think it's actually sending the message quite well, is the comparison between these two birds. So this is a normal, regular bird. It has feathers. This is a poor bird which, due to illness, lost all its feathers. So this is the polypeptide backbone of protein. This is the full protein. And the majority of proteins are glycosylated. And anybody doing science only on a polypeptide or a gene part of a protein is actually studying a model like this. And yes, you can do good science on this type of a model. You can study eyesight, you can study physiology, you can study skin, you can even study this bird walking. But the concept of flight cannot be understood on this model. So if you are studying a model which is missing some of the key components, like the feathers on a bird or glycans on a protein, you simply cannot understand the function of that protein. And I think this is the message we have to send to the global community. You are studying a featherless bird, you cannot understand the model that you are working on. And especially if we are talking about the cell. So the lipid bilayer, the cell membrane, is this tiny little white lane which we see here. This is the lipid bilayer. And the real cell membrane, the real barrier between the cell and the environment is the glycocalyx, this thick coating on the surface of a cell. So you cannot understand anything which is going on between the cells or between the organisms if you ignore glycans. And one of the key elements, which also part of our community often ignores, is that these two components of a protein have a very different logic behind them. So polypeptide part is encoded in a gene. We have a gene sequence which is coding for amino acids, and this is something which is more or less hardwired. It's an evolutionary wisdom through millions or billions of years. It changes extremely slowly. It takes a random mutation and then selection in the next step to change a gene. The other part of a protein, the glycan part, and this is the Fc domain of IgG, the glycans are part of the structure. They're part of the function. So the function of this molecule is partly defined by a polypeptide and partly by glycan. But the glycans, they don't have their genes. Glycans are encoded in a dynamic network of dozens or hundreds of genes. An alternative glycosylation, putting one of these two glycans on a given glycosylation site, is functionally analogous to coding mutation. It changes structures of protein, it changes function of a protein. But it's not being inherited in a Mendelian way, meaning you have a mutation which goes from parent to a child, but it's being inherited as a complex trait, as a number of polymorphisms and all the regulatory elements in this large network of genes which regulate, and this is a network of genes which regulates IgG glycosylation. And we rather clearly demonstrated it 5 years ago. Unfortunately, it went unnoticed by a large part of the scientific community. And this was a study of a collaborative cross cohort of mice. So the mice which was generated by crossing the original parental strains for 3 generations and then inbreeding the offsprings, and practically generated strains from cousins. So they were cousins, but in multiple copies, strains. And some of these strains were structurally and functionally completely separated based on their IgG glycome composition without a single mutation. So the variability between these mice was not caused by mutations. It was caused by different segregation of allelic variants, which were then, of course, fixed because we had copies, the strain of the same individual. And this is also what we see in humans. But in humans, we don't have copies of each of us. But some of us will be here, some of us will be here, some of us will be here. We will be very different from others because we have a different set of allelic variants which define our glycome composition. So once we started to define the Human Glycome Project, we set our mission very widely. We said we want to define structures and understand functions of human glycoconjugates. And at the moment we were defining Human Glycome Project, we said, okay, Human Genome Project was something like putting man to the moon. Once the Human Genome Project started, it was really difficult to sequence genes, and it was not easy to sequence the human genome. But now, you pay 150 euros to Dante Labs to get your own genome. So genome today is easy. And then we said Glycome is way more complicated. Glycome is like putting man to Mars, which was kind of an impossible mission when we were talking about it 7 years ago. But in the meantime, this guy came pretty close to putting man to the Mars. That's not unimaginable. And I really believe that in a couple of years, we'll have man walking on the Mars. And the Human Glycome Project is also in the same path of delivering on our promise. For example, one of the initial goals we made was we want to sequence 100,000 human glycomes. 7 years ago, this was a little bit crazy. Nobody was doing it. Nobody was thinking about it. But actually, last year, we crossed 200,000 human glycomes. So this review paper published last year in Chemical Reviews listed 191,000 human glycomes, which were published as a part of large epidemiologic and clinical studies. And this was largely driven by my lab. Genos generated 159,000 of these glycomes. Manfred was the second largest, and other people generally do not follow this path of generating large amount of data, but they really encourage more of you to start generating more glycomic data, because technology is here, interest is here, and there is clear discoveries coming from analyzing a large number of glycomic data. Because what we did not analyze any 200,000 people. We analyzed 200,000 people from some of the best phenotype, genotype cohorts all around the world, in collaboration with some of the best researchers all around the world. So you could merge glycomic data and all other data which is available. And of course, the easy thing is, let's merge it with genetic data. And we did publish a series of GWAS papers, largely led by Yuri. Yuri is here. He'll have a presentation overview of the 13 years of GWAS studies tomorrow. And today we'll have presentation by Azra, who is somewhere here. Azra, she will talk about what we are doing at this very moment. So at least for IgG, we have largely mapped a network of genes which seem to be regulatory for IgG glycosylation. But the GWAS is a hypothesis-generating tool. It generates hypotheses which genes may be involved. We have to check whether they are or not involved. And this is something what is being done by Lauc and her team. Lauc is not here today, but Annika will have a presentation later this morning to present how they're using their recently developed CRISPR tools to upregulate, downregulate specific genes and show which of the genes that are GWAS hits are actually directly involved in IgG glycosylation. And of course, science is fun, but to see the real benefit of science, we have to move it into the real world, and people have to start using it. And glycans are moving into the real world thanks to the glycan age, which is our spin-out. And a glycan age test of biological age is currently the leading navigator for healthy aging. There are over 1,000 clinics all around the world which are using this test to show whether something what people are trying to do with their lifestyle actually works for them or not. And it was featured in all major newspapers. And general idea is current healthcare system is so-called intermittent healthcare. If something gets broken, we go for help and try to fix it. And imagine if we would treat our cars in that way. Imagine that we change tires only after they explode on a highway. Driving would be very deadly because you don't wait for something to break down to fix it. You try to prevent cars exploding on a highway by doing regular maintenance. We don't do it to ourselves. We run through life until something falls apart. And when something is not working, when we feel pain in one part of our body, we go to hospital and tell to the magicians, fix it. It does not work that way. especially if magicians get paid to treat you all the time. So our current healthcare system is something gets broken, you enter the healthcare system and you become their customers for the rest of your life. You continue pay to sustain your life with much lower quality of life. But the disease is not 0-1 model. It's a long, continuous path of first some molecules changing, then losing homeostatic condition in some specific system, and then widening it to the organs, and then finally organs falling down. And what we are trying to do is to identify changes which happen here. So which is still reversible, which is still not doing a permanent damage, and if you identify them, if you fix them, then your tire will not explode and you will not end up dead on a highway. And glycans are here extremely important because, at least for IgG, glycans are functional effectors with multiple roles in balancing inflammation. And what we know from correlation studies, as we are younger, we have more glycans, which are larger, galactosylated, sialylated. They correlate with the less inflammation. And as we are getting older, there are more and more glycans which correlate with higher inflammation. And this is partly genetic. Specifically for the glycan age, it's 40% heritable. This is something what we got from our parents. This is something we cannot change. But 60% are our decisions. Partly written in epigenetics, which is our lifetime experience from prenatal until now, and partly by the metabolic constellation, which is caused by diet, physical activity, stress, and so on. And these things integrate into glycan structures, which affect inflammation, and then which have effect on future disease. And through a series of studies, and we have Olga Demler here who will present later, we have shown, for example, that for CVD, glycans today predict what will happen, what will be your risk for heart attack or stroke in the future. You know, heart attack and stroke, this is the major killer. And actually, your glycans can tell you whether you're in high risk or low risk. And compared to the genetic risk, where you cannot do anything about it, because if somebody tells you your polygenic score is 4 times higher risk, whatever you do, it will always be a 4 times higher genetic risk of anything. Here, you can actually change your lifestyle, change your risk factor, and see that by doing something, you have decreased your real risk. We know that in medicine, up to 80% of the smart drugs, the expensive drugs, are wasted. They're given to people who do not work, in whom this drug does not work. And then you have to change the drug and change the drug and so on. And we all know, if you have hypertension, you get one drug and then it doesn't work, you get another drug, it doesn't work, and so on. And idea, for the moment only idea, because nobody's really supporting it, is that you have to stratify people and then based on this companion diagnostics, give them the drug which actually works. But something which it's not widely appreciated is that the same thing is valid for lifestyle, diet, physical activity, stress, sleep. We are all very different. Some people can cope with a high level of carbohydrates. The other people cannot. Some people can have a very low carbohydrate, high keto diet, and be fine. Some people will not be fine. We are all different. And one big part of these differences are glycans. Glycans are one of the key molecular sources of inter-individual differences. Starting from the blood groups. Blood groups are chemically glycan. So whenever people ask me, is there an application of glycans in personalized medicine? I say, yes, blood groups. The first biomarker in personalized medicine is actually a glycan. And there are more and more initiatives. For example, Interven is one of the leaders in the field who is developing more and more glycan biomarkers which could be used in patient stratification and diagnostics. Pharma industry has concluded that glycoengineering has to be one of the core elements Most of the new protein drugs involve at least some parts of glycoengineering. Fantastic example is the rituximab. Rituximab is one of the mostly used monoclonal drugs. It's anti-CD30 monoclonal antibody. But ublituximab is 100, 500, maybe 1,000 times more potent. And the only difference is it's glycoengineered. This core fucose, this safety switch, which is blocking ADCC, was removed. And once you make a properly glycosylated antibody, you have a drug which is at least 100 times better than if it's wrongly glycosylated. Unfortunately, still, large part of scientific community, our colleagues at universities, they ignore glycans. Even the director of NIH, at the beginning of pandemics, was tweeting about the S-glycoprotein, glycoprotein, without a single glycan. So he was basing his research on this featherless bird and completely ignoring the glycan structures. Pandemics actually helped. People started to realize the glycans are important. Actually, glycans got even the centerfold of the New York Times. Also, there was a Nobel Prize in chemistry in the glyco field last year, so there is really a strong push toward accepting the importance of glycosylation. There are more and more special issues in the prominent journals, like the chemical reviews, which Sabine mentioned, somehow managed to lobby. Thank you, Sabine. Or the engineering, which Wei was arranging, where we finally managed to put the glycomics as special issues in highly prominent journals. Of course, the Essentials of Glycobiology, a new edition, which is today completely freely available. So if anybody needs to learn something about the glycomics, send them the link. There is a full version of Essentials of Glycobiology. But more and more popular science books are also mentioning glycans. In his crazy popular book, the Food for Life, Tim Spector is mentioning glycans several times in the book. But we have to do more. So recently, a Human Glycome project launched a YouTube channel. I decided to put significant resources to try to generate the content and to promote the content. We already have over 500 subscribers to that channel, which is not bad for a channel which is here for 2 weeks. And I do urge you to help us to promote it, to promote it to your students, to your colleagues, because we'll be putting more and more material there. We have all lectures from the second Human Glycome project meeting. We have all lectures from the third Glycome project meeting. We'll have all lectures from this conference. We are also adding lectures which are more popular, promoting science to other disciplines, and also more about the biopharma. And there are other initiatives. Glycan Age has launched the Glycan Hub with some of the prominent glycobiologists presenting, like Salome Had, Peter Joshi, and they'll continue to do that. I really feel sad when I see that the effort of creating such a large podcast then ends up with only a few hundred viewers. So we have somehow to push it forward. But not only the Glycan Hub, there is the Carbohydrate Chronicles, which is another more carbohydrate-oriented podcast, again, without significant visibility. So this is one of the ideas behind the Human Glycome Project. We try to coordinate our activities in promotion, in education, and in this way kind of spread the gospel in the other communities. I was talking a little bit more than I was supposed, but we are missing one of the lectures, so this first session is more relaxed. And I will soon give my the podium to Chelsea, who is the head of the chairman of this first session. But before that, I will remind you again, we are recording. So presentations are being recorded, questions and answers are being recorded. If anybody has problems with that, please let us know in advance, because I think it is important to have the lectures presented to the wider audience. If somebody disagrees, we will not do it, of course. We will not publish anything without your consent. But if you give us the copy and you do not say that you are not you do not agree with the presentation, with the publication of presentation, then we will assume that you agree. So thank you again for coming, and Chelsea, please take over with the first session.